In this video, we will discuss about a Ras inhibitor compound, that is RMC-7977, that exhibit anti-tumor activity against pancreatic cancer cells in vitro. So we will start a discussion with the panel A. In the panel A, they check the viability of different cancer cell lines against RMC-7977, and they have find out that compared to other cancers, the pancreatic cancer cells has low viability. That can be seen in this panel A, where they compared other cancer cell lines and also the pancreatic cancer cell lines, and they have found out that pancreatic cancer cell lines has low viability. And thereafter they check the sensitivity of the human PDAC cells against different RAS mutations that are shown on the horizontal axis, and they have find out that the sensitivity of this RMCC is high to some of the most common RAS mutations that are found in the human PDAC cell lines. That is G12X, that is G12 get mutated with DVCR. So here the sensitivities are higher, so indicating that the RMC is more sensitive towards the human most of the human PDC cell lines, as most of the human PDC cell lines has the RAS mutations at around G12X position. And thereafter they check the human PDAC cells of the sensitivity against the two different kind of RAS mutations, and they have find out is that for RAS G12C the viability test. So what they have find out is that viability at this RAS G12C is low compared to this RAS G12D. And when they check for the human PDAC organoid, they have find out is that for both the conditions the sensitivity is almost similar. No sensitivity, viability of this PDAC organoids are almost similar for both the kind of mutations. And then they wanted to check what are the actual reasons that are occurring. So they think that this phosphorylation of these PERK pathways or the S6 pathways might be triggered when this RMC interact with this these HPAC cells. So when they check the HPAC cells, they find at different concentration of this RMC-7977, they find out that there is a decrease, there is a decrease in the phosphorylated ERK pathway and phosphorylated PS6 and PK phosphorylated AKT pathway, which is the survival pathways. There is a decrease in the survival pathways, indicating that survival capability of the cells with increase in the RMC concentrations goes down. So once the survival capability of the cells goes down, the hypothesis that the apoptosis pathways or capability might goes up. So in the human HPC cells, they check the apoptotic factors such as this C-PARP and this CC3 are not shown in the Western blot. So in the C-PARP, they find out that after 48 hours of this 100 nanomolar RMC-7977 injections, they see the increase in this C-PARP, which is the indications of the apoptotic in the cells. So once they find out that apoptotic factors are increasing, they wanted to check this behavior in the human PDAC cells. So what they take the tissue slides of the actual human that is infected with this PDAC cancer cells and test them using these different concentrations of the RMC-7977. And what they find out is that with increase in the concentrations, they see the decrease in the phosphorylated cells per field. That is, when RMC-7977 is 0, the PERK signaling pathways is high. However, with increase in this RMC-7977, they see the decrease in the PERK signaling pathways for different kind of RAS. That is for the RAS wild types, RAS G12V and RAS G12R. These are the two different kind of mutations that are found in that donor patient. And after that, they also check simultaneously. They also check the apoptotic factors such as CC3, which indicates that apoptosis are there in the cells or not. And they find out is that with increase in this RMC-7977, they see the increase in the apoptotic factors that can be seen here in the panel H. That with increase in the RMC-7977, the CC3 cells per field increases, and the IHC images are shown in the panel I, where we can see that for these persignaling pathways with the RMC-7977 treatment, the persignaling pathways goes down drastically between the compared DMSO. And for the CC3 signalings compared to the DMSO, we can clearly see the increase in these CC3 signals when treated with the tissue slides or treated with RMC-7977. This indicates that the RMC-7977 is a exhibit. RMC-7977 exhibits some anti-tumor activity against the PDAC cancer cells from this experimental results. So I hope you understood how they come up to a point that RMC-7977 exhibit anti-tumor activity against PDAC. If you need to understand more about these experiments in detail, you can go through the reference that is provided in the description. So that's all for this video. Thank you for your time.